affinity chromatography is one of the most powerful chromatography technique. So in this case, we can basically it is called single step purification technique because of the high affinity and very high specificity of the ligand to its binding partner. Okay, so here we can do affinity chromatography based on natural affinity of biomolecules to each other, or we can also introduce affinity by putting affinity specific tags. Okay, so both are possible. So in case of natural affinity, can you give some example of biomolecules having natural affinity for each other? Any example of biomolecules having natural affinity for each other?
The polar
Agent,
molecule.
What?
enzyme, substrate,
Enzyme substrate. Yes, very good. Anything else?
affinity, biotin,
Avidin, biotin. Avidin,
avidin,
biotin,
biotin,
streptavidin, biotin. Yes, very good. Anything else?
antigen, antibody.
Yes, antigen, antibody. Very good. Okay. So these are some natural affinity between biomolecules. Also, we can introduce artificial affinity by recombinant DNA technology by introducing the affinity tags. Like yesterday we saw MIG tag, HIS tag, MBP tag, flag tag. All those tags can be introduced by recombinant DNA technology to introduce artificial affinity. Okay, so both can be done, but in any case, this is a very powerful technique. So it was first developed in 1930s uh, by Wilhelm Silas, who was a Swedish biochemist, and he was awarded a Nobel Prize for this in 1948. Initially, it was used to study enzymes and proteins and their receptors, all those things were initially used. And basically, as the name suggests, it relies on the affinity of various biochemical compounds, biological molecules with specific properties. Okay, so that is why it becomes very, very specific and very strong tool for protein purification. This is just an example with the artificial tag. So here you can see this in this case a GST tag, okay, which is glutathione S transferase tag proteins. Here this is the glutathione S transferase tag protein, and I was mentioning yesterday to you that sometimes after purification, if you want to get rid of the tag, that also can be done by recombinant DNA technology. So in this case, you can see this is our protein of interest, which is tagged with GST, and in between you can see there is a thrombin site. Okay, this is a protease site. So once you are able to purify your protein, you can use this protease to cut here, and the protein will be separated from the tag. So basically, how here it is working, you see that glutathione का substrate है glutathione S transferase का substrate है glutathione. Okay, so this is the glutathione, you know, this is a glutathione का this is 3 amino acids से बनता है. So this is a glutathione uh, and this glutathione is covalently linked to the porous bead matrix, which can be agarose or saccharose, but it is covalently linked. So covalently linked होने से this cannot be just washed away from the column, and so the column is formed, stationary phase of the column is formed of these beads having glutathione covalently linked to the matrix. Okay, and to this column, when we add a mixture of protein, amongst the mixture of protein, one of the protein is our protein of interest, which is having a GST tag. Okay, so this GST will bind to glutathione, but since this glutathione is not free, it will not be leaving the enzyme. Okay, so this will be attached here, and all your protein which is having the GST tag will remain in the column, while all other proteins without the tag will just be removed by washing. Okay, and when you have to elute your protein, there are two ways. Either you can use a competitor in this case will be free glutathione. You add excess of free glutathione, that free excess of free glutathione will compete with bound glutathione, and thereby protein will be eluted out of the column. But in that case, the eluted protein will retain the tag. Okay, it will not remove the tag. But if you want to remove the tag, then you add the enzyme. The thrombin is a protease, so you add thrombin protease in the column. That protease will cleave this at this site, and this GST will remain bound in the column, and your protein only will come out in the elution. Okay, so this is, in this way you can get your protein without the tag. Is it clear to everyone what I'm saying?
Yes, sir.
So as you also told, these are some examples of natural affinity: antigen to antibody, antibody to antigen, and enzyme substrate, DNA and histone. Do you know what is histone? Histone क्या होता है?
Histone protein in binding up of
Histone
DNA,
proteins.
sir.
Yeah, these are proteins on which DNA is wrapped around in the nucleosome conformation. So histone proteins have very high affinity for DNA. Same we have hormone and the receptor. Okay, so these are all examples of natural biological affinities. Okay, so all this can be utilized for affinity purification. Okay, so basically, as in other cases, these are the basic requirement for chromatography. We should have a matrix which is uh, required for ligand attachment. The ligand can be attached either directly to matrix, or sometimes we can put a spacer arm which will provide some distance, and to this spacer arm, then our ligand, which is the molecule for the specific target molecule for interaction, will attach. So in the previous case, this ligand was glutathione. Okay, so this glutathione is attached to the matrix via spacer arm. Okay, so you make a affinity resin this way, and then that can be used for purification. Okay, so normally once you have the column, the sample is injected in the column in the buffer, which is already column is equilibrated with the buffer of choice, and only the substance with the affinity for the ligand will bind to the column, and others will be eluted off. And as I just said, the substances which are retained in the column can be eluted by either changing the pH, or you can use a competitor, or you can use a protease. In any of these ways, you can elute the bound protein from the column. Okay, and as affinity is very specific to your protein of interest, there will be very less chances that non-specific binding will be happening, because if you wash it properly, your protein, in most cases, you will get a pure protein in one go. So matrix normally, as previously shown, normally it is a uh, matrix made up of agarose or saccharose, and it will have certain biological active functional groups like amino group or hydroxyl group or carboxyl group, so that it can act as target for attaching your linker to it. Okay, and that linker will then be further attached with the ligand. Okay, so also another property of the matrix should that it should be reusable. Okay, so once your purification is done, you can wash the matrix with either sodium hydrochloride or hydroxide or urea to remove any uh, clogged proteins or precipitated proteins from the column, and then it should be ready to be used again. Okay, so matrix should be reusable, otherwise it will become very expensive. Okay, so ligand can be any molecule which you are having a specific affinity with your target molecule. Okay, so that ligand can be a antibody or antigen, or it can be DNA. Okay, so that ligand is the one which is you are targeting your protein for purification. Okay, so accordingly you put the ligand in the complex. So you have to covalently link the ligand to the spacer molecule, spacer arm of the column, and then the ligand molecule complex will be remaining in the column, eluting everything else. And once the ligand molecule complex is washed thoroughly, then you can dissociate and purify your molecule of interest by changing pH, or by using a competition, or by adding a specific protease. Another example of affinity purification using natural affinity is purification of immunoglobulins. Okay, All of you know what are immunoglobulins?
Yes, sir. Antibody.
Antibodies.
Yes, antibodies. Okay. So antibodies, if you want to purify antibodies, apart from antigen, they also have high affinity for some certain proteins. In this case, there are two proteins called protein A and protein G, which are proteins from Staphylococcus and Streptococcus bacteria, and they have very high affinity for the constant region of IgG antibodies. Okay, so if you want to purify IgG antibodies, you can put either protein A or protein G, attach them to the matrix through the linker, and when you pass the sample, you can potentially serum sample or where you want to purify antibodies from. If you pass through the column, those antibodies will bind to protein A or protein G, and thereby after washing the column, you can change the pH. So by changing pH, the affinity is reduced and the antibodies can be eluted out of the column. Okay. So as I said, this protein and protein G are bacterial surface protein from Staphylococcus aureus and Streptococcus bacteria, and they are extensively used for purification of immunoglobulins. If you don't have natural affinity in your biomolecule, then also you can use affinity chromatography. As I said earlier, in that case you have to make use of recombinant DNA technology, wherein you can introduce artificial affinity tags in your proteins. So these are some of the popular tags: HA tag, flag tag, his tag, MIG tag, and the amino acid sequence which codes for these tags. So based on this sequence, corresponding DNA sequence you have to introduce in your vector, and then your protein will, whatever proteins you produce will have this particular tag in them, and then you can use this. So for example, for HA tag purification, in the column you have to put anti-HA antibodies. For flag tag purification, you have to put in the column anti-flag antibodies. For his tag purification, this his tag is nothing but, you can see there are 6 to 8 histidine amino acid in a row. Okay, continuous. Here it is showing 6 histidines, so 6 to 8 histidines can be put in a row. That becomes a his tag. Okay, so in case of his tag has very high affinity with a metal ion called nickel. Nickel सबने सुना nickel? Have you heard of nickel?
Yes, sir.
So histidine has it forms coordination bond with nickel. So in this case, the column is having the nickel metal, and through that interaction, these histag proteins are uh, purified. Also, MIG tag. This is a sequence of MIG tag. If you are putting MIG tag, then in the column you have to put anti-MIG antibodies. Okay, so thereby artificially also you can create condition for affinity purification of proteins. Okay, so here this is, I was just saying, his tag is the most common tag because it is also the most cheapest tag to be used. So normally his tag proteins purification putting his tag is the most common way of purifying proteins via RDT. Okay, so there are several vectors commercially available. This is an example of a PAD28A plus vector which is utilized for the expression of proteins in E. coli system, and in this systems, the histag sequences are already present in within the commercially available tag. Here you can see, here it is histag coding sequence here. So between 140 and 157 base pairs, histag sequence is there. Okay, so also there you can see there's another sequence. So you can have histag either at the N terminal of the protein or C terminal of the protein. So here also there is another histag protein sequence between 270 and 287 residues. So basically, if you clone genes in such vectors, you have proteins which are produced will have his tag. Okay, and then that his tag, as I said, this is how this uh, histidine is able to form a histidine nickel complex. So you can see here there is a coordination bond between nickel and the histidine nitrogen. Okay, and this is how the proteins containing histidine they are bound with the column which has nickel on it. Okay, and then to remove it, we use a molecule called imidazole, which has similar this ring of histidine is called imidazole ring. So we use a free imidazole, and when we put free imidazole, this is able to replace the his tag proteins from the column, and imidazole will go and bind to the nickel. Okay, so by providing competition with imidazole, you are able to you are able to elute the proteins out. Okay, so normally when you are using E. coli systems, I think most of you might have done it or must be aware that you have to transform your gene of interest with the his tag in this into clone into PET28 vector. In this case, you have to then transform into E. coli uh, and E. coli strains which are meant for expression of protein, like in this case E. coli BL21DE3 strain. Then you select for the clones using the antibiotic marker. You can see here uh, in this case uh, you have a kanamycin resistance marker here, so you have to select with kanamycin antibiotic. Then once you have the right colonies, you grow them, and normally when the cells
वो सब आरडीटी कोर्स के लिए रहने दीजिए, और क्या है
uh,
सब
okay,
आरडीटी
okay. If they are aware, then it is fine. Basically, you normally use this system for expression of protein and IPTG induction of protein, and then once
Yes.
you have enough protein, then you have to sonicate. As I said, we have different methods to lyse the cells. So here normally we use sonication. So after sonication, your soluble proteins are there in the supernatant, and to the supernatant you add your nickel resin. Okay, nickel NTA resin you put. So all the proteins which are having histidine tag, they will only bind to the resin. Rest of the column, rest of the proteins will not bind, and they will be washed off of the column. Once you have all your protein of interest bound to the column, then in the elution buffer you can add. You can, if you are doing first time, you can add increasing amount of free imidazole. Here in this case, I am writing 100 to 400 millimolar of free imidazole, and then after incubation by competition, your protein of interest will come off from the bead, and imidazole will bind to the column. Okay, and then you can take out the samples, run on the SDS page, and followed by coma staining, so you can see which fractions have your protein of interest in them. Okay, so his tag purification, his tag is one of the cheapest tag as well as one of the cheapest method for affinity purification. Whenever you are using recombinant DNA technology, the easiest way to use is his tag and nickel NTA chromatography. So here this is just an example. This is again from my lab only. So you can see this is the starting material. Can you see this? How many bands are here? And this is the one protein of our interest. And after subjecting this many proteins to nickel column, you can see that only our protein of interest is purified. All of you are able to see this?
Yes, sir.
So this is the power of affinity purification. You do not need to do any initial ammonium sulfate precipitation or any other step. Directly you put this mixture of thousands of protein in the column, wash it nicely, and then dilute with free imidazole, and you get your protein of interest, pure protein of interest. Okay. Okay. Now next chromatography technique which we are going to discuss is hydrophobic interaction chromatography. All of you know that there are amino acids which are hydrophobic in nature. Can you name some of the hydrophobic amino acids?
Excuse me, sir.
Yes.
Sir, in affinity chromatography, suppose we have the substrate, any substance, but we do not have any specific ligand for it. So in that case, how do we can do that?
No. Can you say, for example, I just told you, na, if you don't have ligand, then you have to artificially add a tag to it. That is what you do.
Sir, that is work for the protein. Suppose we have some other substance, not like that. If any other polymer is there, which is we need to separate it out. In that cases, how to do that?
Okay. So in that case, yeah, you know, tag, these tags may, as you said, these tags are all peptide based. Okay, so these tags are uh, good only for proteins, but basically, in that case, normally small molecules you can tag with biotin. Okay, biotin is a smaller molecule, so biotin streptavidin kind of affinity uh, can be created. But again, it is difficult because tagging specifically to one molecule, it is going to be difficult. So I think I have not focused on that line because we work only on proteins. But that is a good question. But small molecules like biotin can be potentially used instead of a peptide tag.
Hmm. Actually, sometimes, sir, we do not have much idea on that. Some substance when we isolate and we want to purify it. So in that time, this ligand preparation for that to separate it out is sometimes it is very difficult.
See,
So that's
another way is like to use some bioinformatics tools to know that what can potentially interact with your molecule of interest. Okay, so sometimes bioinformatics might help you to identify a potential binding partner for it.
okay.
Also, normally whenever you have this kind of, also, in this small molecules are there, instead of this FPLC, HPLC is more preferred. Okay, so HPLC is generally good for smaller molecules. Proteins are generally large molecules. If you have smaller molecules, HPLC is a better way to purify. So what you do is, if you have some rough idea about the properties of your molecule, and then if you have a diagnostic way to know that in which fraction it is going to come out, if you have a test for that, then you can first run an HPLC column and try to see that how it behaves on the column, what is the retention time, all those things, and then it will potentially help you to finally fine tune your strategy to purify it.
Okay. Okay. Thank you, sir. Thank you very much.
So this what is hydrophobic interaction chromatography is basically mostly used in HPLC only, as we will see now. Okay. So I was asking that, can you name some hydrophobic amino acids? Anybody?
Sir, mainly the
Glycine,
aromatic amino.
Aromatic are all hydrophobic.
Not all tryptophan, tyrosine.
Hmm.
glycine.
Lysine is hydrophobic. Lysine is not hydrophobic. It is hydrophilic.
Glycine, sir.
Like
Leucine,
leucine, isoleucine, valine.
isoleucine, alanine.
ये सारा ये देखो ये सारा ये these are the hydrophobic, 9
Alanine.
amino
Yes,
acids
sir.
are hydrophobic. Okay, glycine, alanine, valine, leucine, isoleucine, phenylalanine, methionine, and tryptophan. Okay, proline. Okay, so these are the hydrophobic amino acids. So whenever your protein are rich in these amino acids, and if these amino acids are present on the surface, then the hydrophobic interaction chromatography is a good tool to use for purification of such proteins. Okay, so how it works, let us see. So normally this hydrophobic interaction chromatography is used as a downstream purification. Somebody was yesterday asking me that after gel filtration or ion exchange chromatography, if there are still some molecules which are, or some proteins which are present as contaminating proteins, what can be done? So this is one way you can exploit the hydrophobic interactions to further separate them. Okay, so that's why it is called a downstream purification technique. So just excuse me, let me do. So it is normally exploiting differences in hydrophobicity. So the number of hydrophobic amino acids and distribution of amino acids. Okay. So based on this, the two proteins will have a different number as well as distribution. So taking advantage of that, this can be utilized for further purification. So normally, here I am giving example that after an ion exchange, if you have still some impurities, you can go for hydrophobic interaction chromatography. So how it works? So normally, whenever you have hydrophobic surfaces, they are surrounded by an ordered monolayer of water molecules. So for example, this is a hydrophobic surface. So the water molecules will maintain a ordered layer around them. This is the second hydrophobic surface. Again, water molecules will form an ordered layer around them. But if you bring these two surfaces close to each other, what will happen? That either you bring them close, or if you add in this solution, if you add something which is attracting water, okay, which can attract the water molecules. For example, we took example of ammonium sulfate precipitation. When you add this salt in water, this salt is highly soluble in water, so what it will do is it will attract away some of the water molecules. Okay, so when the water molecules are taken away, then the hydrophobic surfaces of this protein, they will come in close vicinity, they will form a hydrophobic interaction, and the water molecules which are less now again will form a ordered layer around the interacting biomolecules. Okay, so by doing this, uh, by minimizing the exposed surface area, this will increase the entropy. Okay, so and the free energy also will be negative, so it will be a favorable reaction. Okay, so this is how the hydrophobic interactions are promoted. So you will see exactly what happens. So here in this case, what happens, you have a column. So in the column, this is again your matrix. On the matrix, this H is your hydrophobic surface. Okay, so hydrophobic surface is put on the column. Okay, so this, when you have your proteins, okay, mixture of proteins added to such columns, what will happen, that at you have to use two salt concentration. At low salt concentration, what will happen, and at high salt concentration, what will happen. Okay, so at low salt concentration means when salt is less, there are more water molecules in the buffer. Okay, so what will happen at low salt concentration, the hydrophobic interaction will be minimal, because the water molecules are sufficient, they will be covering all hydrophobic surfaces as shown in the previous slide, so they will not be interacting. But as soon as you increase the salt concentration in the buffer, the water molecules, free water molecules are taken away, and now these molecules, they have no choice but to be stable, they have to interact with the hydrophobic residues present on the column. Okay, and the molecules or protein molecules which don't have hydrophobic residues in them, they will not interact and they will be washed away from the column, while the proteins which have hydrophobic residues, they will interact with the hydrophobic column and they will be retained. Okay, so this is first binding of the protein with the hydrophobic residue to the column. Now if you want to elute them out again, what will you do, you will again add low salt buffer. So when low salt buffer is again added to the column, again more water molecules are available. The water molecules will again make a layer around the hydrophobic residues, and then again this interaction will be broken and the proteins can be eluted out of the column. Okay, so basically this is column with resin. You load them in the column. So with when you load the column with high salt buffer, hydrophobic residues of the protein surface will interact with the hydrophobic resin, and then when you have sufficient binding of the protein, you elute the target molecules with the low salt buffer. So you can see here, once the molecules have bound, you reduce the salt concentration, and the molecules will come off, and they will come off the column, and thereby you can purify the protein of interest. Okay, ठीक है, तो ये तो ठीक है, we have already discussed. अभी
Excuse me, sir.
yeah.
So that hydrophobic region which is attached with the resin, it is like a common hydrophobic region, like a protein which we are eluting.
No, I did not get your question. Can you repeat?
Sir, I am saying that in the hydrophobic region of the resin which is attached with it, it is like a same of that which is we are.
No, 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 no. I should have told. Okay, so this hydrophobic, you are saying this one, na?
Yes, sir. Yes, sir.
No, no, no. Here, this hydrophobic region is not an amino acid. Okay, this hydrophobic region mostly is a hydrocarbon chain.
Okay. Like for
Okay.
example, sir, what?
For example, it can be CH3, 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 CH2. Okay, so it can be a 4 carbon, 8 carbon, 18 carbon, like this. In HPLC column, if you have used you see that the nomenclature of HPLC column is like that only: C4 column, C8 column, C18 column. किसी ने सुना है?
It is like a glutaraldehyde or something kind of things.
No. These are not glutaraldehyde. These are covalently linked. Okay, so hydrocarbon chain. मतलब C4 chain means CH3, CH3, CH2, 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 CH2. C8 column has 8 carbon. C18 column has 18 carbon hydrocarbon chain. Okay. So as you increase the number of hydrocarbons, the hydrophobicity of the column is increased.
Okay.
So to the resin is same. You can resin in this case most of the time resin is silica. In HPLC the resin is silica based. Okay, to silica resin you add this hydrocarbon chain, so that will make it hydrophobic.
Okay.
Okay.
Fine. Okay. Thank you, sir. Thank you very much.
Okay. So now, as I said, once you have purified the protein, next question is to assess the purity. You have to know that your purification method has worked or not. So you have to assess the purity of the protein. So what are the methods to assess the purity of the protein? One, as we have been talking all through, is you run the samples, different fractions on SDS page, and do the staining, protein staining. So you see that if your preparation is pure enough, you should see only one single band of your protein of interest. Okay, but if you see more bands, that means they are impurities, other proteins are there. Then accordingly you have to change the strategy, modify the strategy, so you can get a single protein. Other way you can check the purity of your sample is, sometimes the protein might look single and pure single band, but if you want to see that there can be some other small amount of proteins which are not detectable by Comasi staining. So what you can do is you can subject your protein sample, the eluted proteins, to mass spectrometry. So mass spectrometry, what it does, that it will make chop your proteins into small fragments using proteases like trypsin, and then they are subjected to LCMS and followed by MSMS, and the sequences which are obtained from mass spectrometry are then aligned to known database of proteins to decipher the identity of proteins in the sample. So if all the peptides are corresponding to your protein of interest, then you are sure that yes, your protein of interest is indeed purified to 100% purity. But if you see that there are other contaminating peptides in the sample, then you can say that yes, okay, there are still some other proteins present. Then you can modify the strategy to remove those contaminating proteins. Another way to check the purity or also confirm the identity of your protein is doing Western blot. So Western blot utilizes the specific antibodies which are targeting your protein of interest. So if your protein is really your protein of interest, the antibodies against it should bind to it and should be able to detect it. So another way to confirm that indeed your protein of interest is the one which is purified, you can subject it to Western blotting. Okay. So normally this is the way in which SDS page works. So how many of you have used SDS page yourself? Anybody? Nobody has used SDS page? Okay, anyway, so this is a method to
बहुत सारे लोगों ने मुझे रिप्लाई किया था.
क्या?
सर प्रोटीन सेपरेशन के.
हाँ, इलेक्ट्रोफोरेसिस सुना ना, इलेक्ट्रोफोरेसिस पॉलीएक्रिलामाइड जेल
हाँ सर
इलेक्ट्रोफोरेसिस. हाँ,
सारे
तो
चीज मैंने बता दिया है, वो तो 10-12 टाइप का बता दिया है.
so then you should tell me then. Okay, so here this is just, and then I should not waste much time here. So you can see in SDS page we are using SDS which will bind to the protein and will give a uniform negative charge. And also we had a disulfide bond break करने के लिए we had a reducing agent like beta mercaptolithanol or dithiothyretol. So the proteins are in linear form. We are boiling them, running on the gel. They will separate based on the molecular weight. And here is an example when you subject your protein for purification technique. your protein of interest, which was initially present with hundreds of contaminating bands, eventually you can get them as a single pure band. Can you see here that from so many bands, your protein of interest could be purified as a single band? Visible to everyone?
Yes, sir.
So this is how your technique should work. You should be able to purify your protein of interest from thousands of protein, and you have to really standardize it well so that you get really one protein of interest nicely from it. Okay. So once you have your protein of interest, also you might need to have its quantification. Okay, so there are different ways in which you can quantify your protein. So there are several spectrophotometric methods in which the proteins can react with different uh, components, like for example biorate assay, in which your peptide bonds within the proteins will bind to the alkaline copper sulfate, and it will produce a purple color compound which can be read at 540 nanometer. Then it is a modified biorate assay, which is a Lowry assay, so in which the purple compound from biorate assay is further reacting with a Pauling et al. 2 reagent, and it will produce a deep blue color which can be read at 650 nanometer. And this is sensitivity from 1 milligram is further increased to up to 5 microgram. Then there are Bradford assay, in which Comasi brilliant blue, the dye binds to the positively charged residues of the protein, and thereby it also aids in quantification of the protein. And here also this is a sensitive method. You can from 1 to 20 microgram of proteins you can estimate by Bradford. Then you have spectrophotometric method, the absorbance at 280 nanometer. Also you can sometimes do, this is not routinely done, but amino acid composition analysis can be done. But here you have to destroy the protein and uh, chop them into individual amino acid, and then, but this is normally not done routinely, but these are the common methods which are utilized for quantification of the proteins. Okay. Then also, if you want to use the purified protein for sequencing, then there are methods for it. So first method is called Edman degradation and sequencing, and secondly, we can do it by mass spectrometry. So we'll quickly go through how they work. So for Edman degradation of protein and the sequencing, normally first the protein is cut into smaller peptides of around 50 amino acids by using endopeptides like trypsin. So trypsin, normally all proteins have trypsin cutting sites. So using limited proteolysis by trypsin, you first get small fragments around 50 amino acid long, and then these peptides are put onto a solid glass fiber matrix with a cationic polymer, so that because of the ion-ion interaction, electrostatic interactions, the peptides will be attached to the solid matrix, and then you add the Edman degradation reagent, which is phenyl isothiocyanate in alkaline condition. So what it does, that PITC reacts with the N-terminal amino acids and derivatize it. Okay, and this derivatized amino acid is then it is released from the peptide sequence by using anhydrous acid, and this released uh, then amino acid is identified using HPLC. And this I'll show in the next slide how exactly by the cartoon how it is happening. But this cycle is then repeated one by one. So every time the N-terminal amino acid is derivatized, it is removed and then identified. Okay, so this is how it works. So you see, this is a sequence of protein, like normally 50 amino acid long protein. So the N-terminal residue of this peptide is derivatized by PITC, and this derivatized molecule is a N-terminal is then by using anhydrous acid, this is removed, and this removed derivatized amino acid is passed through HPLC, and HPLC will be able to identify what it is, and one by one each amino acid is cut and the sequence is put together. So you know, that way you can identify the entire sequence of protein. Okay, so this is advanced sequencing, but nowadays we have advanced methods. So advanced sequencing is lesser used. What we now normally use is mass spectrometry. So mass spectrometry, how it works, so here again you can have a mixture of proteins in which you want to identify the proteins. So what you do is you again digest the proteins into small fragments. Again, here also trypsin and chymotrypsins are the enzymes which are utilized. So they will make smaller fragments of the protein, and the smaller fragments of the protein are then passed through HPLC. So HPLC, based on the size as well as hydrophobicity of the peptides, they will be separated out, so different peaks can be visualized. And once these different peaks are coming out of the HPLC, they are directly fed into mass spectrometer. Okay, so mass spectrometer have uh, initial initial part as ionization, so all these peptides are initially ionized. So based on the ionized form of peptide, they will have a mass to charge ratio, and thereby in the mass spectrometry you get a peak based on their charge and mass ratio. And then these peptides can be further in the, whenever there is a tandem mass spectrometry, then when there are two chambers in that, first will uh, record the native molecular weight of the mass to charge ratio of the native peptide, but then it can further create further fragments of this peptide. So you can see that when the further fragments this is the entire peptide, and smaller fragments will have a different peak because their mass to charge ratios are altered. And uh, this particular pattern is compared with the database. Okay, database of proteins and peptides, and based on the uh, mass to charge ratio of the original peak, and as the daughter peaks. Okay, you can combine them together to derive the sequence of the peptide. Okay, so this is now more, mass spectrometry is more commonly used to determine the sequence of the proteins. And also good part is that here you can use a mixture of proteins at one go. And as I was telling in the detection part also, if you want to see that your protein is 100% pure or not, so if your protein is 100% pure, your all peptides will belong to only your protein of interest. If it is contaminated with other proteins, then you will have other peptides also here, and they will have a different pattern. And you can then determine that yes, different peptides belonging to different proteins are also present, and thereby you can say that your preparation is not 100% pure, and you can accordingly change the strategy or modify the strategy. Okay, once you have purified proteins, okay, one of the major use of purified protein is to determine the structure of the protein. Okay, so there are several labs, several people, researchers are totally devoted to get the structures of different proteins. Okay, so there are different ways in which you can determine the structure of the protein, whether you are interested only in the secondary structure or you are interested in the tertiary structure. Based on that, you can use different techniques. So here we will be briefly telling about the four techniques which are commonly used for determination of the protein structure. First is circular dichroism CD spectroscopy. Mainly it is meant for secondary structures. Then we have NMR, which is nuclear magnetic resonance spectroscopy. Then we have X-ray crystallography and diffraction, and cryo-EM, which is most recent development in the uh, field of structural biology. So normally, all of you must be aware that these are the different structures in which proteins can be present. This is the primary structure, is the sequence of amino acids. Then you have secondary structures like alpha helical structure or beta sheet structure. Then you can have a mixture of them, which can form a tertiary structure. And then quaternary structures, when multiple subunits come together, they can form a quaternary structure. Okay. So when we are using circular dichroism, which is a form of spectroscopy, what you it produces a spectra. Uh, so you have you run a wavelength scan. So you can see here wavelength scan between 180 nanometer to 250 nanometer is run. So based on the pattern, I mean based on the what kind of secondary structure the protein contain, you can get either of or a mixture of this kind of pattern. So you can see that if your protein has uh, maximally alpha helical structure. So in that case, you will see this kind of pattern, that you will have a maxima at 192 nanometer and a minima at 208 and 222 nanometer. Okay, so this kind of pattern, if you see, that will tell that your protein is basically alpha helical. But if your protein is rich in beta sheet structure, you will see a maxima at around 195 nanometer and a minima at 280 nanometer. Okay, and always you can have a mixture also. Okay, it is not that it is a pure alpha helical protein or a pure beta sheet containing protein, but if both are present, then both these features will be seen. Okay, and also if it is a random coil, then this kind of structure will be seen. Okay, so using circular dichorism and using the known behavior of the protein, that if what kind of content is there, you can compare with this spectra, and that will give you idea that what kind of secondary structures your protein is forming. Okay?
Excuse me, sir.
Yeah.
If the substance is same or compound is same but in different structures, so that time also it will give a differentiation like this, or
No, I am not getting your point. If compound is same,
suppose there is same protein is there
uh,
that is available in two different structure in alpha helix and beta sheet, and
okay.
both is a mixture. So that time it will CD can differentiate it.
Yeah. So if a protein has a mixture, as I shown here, here
So
you can
same
see
protein, sir. There is
same,
no difference in the protein, sir. Only
same
the
protein. Yeah. This is, you see, this protein has, it's a mixture of, there are beta sheets also and alpha helical structure also here. You can see.
uh, that I can see, sir. My question is that the protein is same, only structure is different. There is no change
Yeah,
in the protein.
no. That is what I'm saying. A same protein at different location within a same protein, you can have amino acid folding into alpha helical structure, and at some other location it can form beta sheet.
Okay. So that is
So
possible.
if there is a mixture of secondary structure, doesn't mean that your protein is entirely, it should be alpha helical, or entirely it should be beta sheet. Okay, it can be a mixture also.
Mhm.
So if there is a mixture, in that case, both patterns you will see. Then you will not see only single pattern. Both patterns will be visible. Okay.
Okay. Fine. Okay, sir. Thank you. Thank you, sir.
Okay, so this is for mainly for secondary structure. Okay, now if you want to do a tertiary structure, normally the most common way used is X-ray crystallography. So first for that you have to form a crystal. Okay, so your protein should be able to form a ordered crystal. So once you have a crystal of your protein, then you bombard that crystal with X-rays. In X-rays, all of you know that X-rays will be able to pass through only where there is a space. Okay, it will not pass through the solid part of the proteins. So what happens eventually, that when the wherever the X-ray eventually is put on the detector, they will produce a X-ray. Whenever you expose the X-ray film, also it will produce dark and light. All of you must have seen the X-ray film जो अपन bone के लिए या structure के लिए देखते हैं. So wherever X-ray could pass, those areas will become dark, और जहाँ X-ray pass नहीं होता है वहाँ पे it remains light. Okay, so based on that, here also it's a molecular X-ray only. Okay, so the X-ray are passed through the crystal of the proteins, and based on the diffraction pattern, there are different softwares which will give you the electron density map, and that by fitting using the software again, you can make an atomic model, and using that atomic model, and again here a lot of bioinformatics and softwares are required, but then you can refine it and then eventually come up with a X-ray crystallography or diffraction or 3D structure of the protein. Okay, but again, you should be expert in deciphering the diffraction pattern. You should be expert in some of the bioinformatic tools, and then you are able to give a 3D or the three-dimensional structure of the protein. Okay, so normally nowadays, again, X-ray crystallography was very popular, but nowadays it has been, wherever possible, it has been replaced with cryo-EM, because there is one big limitation of X-ray crystallography, that your protein of interest should be able to form a crystal. Okay, so there are cases where your purified protein, your protein of interest might not form a crystal. So if it is not forming crystal, the X-ray crystallography or the X-ray diffraction method cannot be used for solving the structure of those proteins. So to overcome this limitation, a new technique was developed which is called cryo-EM. So cryo-EM is cryo as in very cold temperature, cryogenic, cryo. You have heard this term cryogenic, okay? Cryogenic engine. So basically a very cold temperature followed by electron microscopy. Okay, so here what it's done, your sample which is a protein sample is put on a grid of metallic grid, and it is frozen at like minus 200 degree centigrade. Okay, so what happens that at this low temperature, uh, even if they don't really form crystals, but they are frozen in such a way that they will retain a particular shape. Okay, so they will be frozen in a particular shape. So under that frozen shape, they are visualized under electron microscope, and the data is collected. Okay, so what that happens that your molecules might have been frozen in different conformations. So you can see that molecule can be frozen in different conformations, but again, based on the bioinformatic tools, eventually average of these all structures is put together in form of a 3D map, and based on the 3D map, then you can uh, potentially identify that if the structure is like this, what can be the way it is folded. It is folded in alpha helical or a beta sheet form or a mixture of that or a random coil. So based on this, you can come up with the model. Okay. So here this cryo-EM gives an advantage that proteins which are not crystallized also can also get the 3D model of them structure. Also, even in one of my research, my own work, we had used cryo-EM. In India, there is only one cryo-EM system right now. I think it is in ISC Bangalore or NCBS. I think NCBS Bangalore. Okay, it's very expensive, but uh, slowly and slowly we might get more instrument in the country. But we, in my postdoc work also, we used cryo-EM to solve crystal structure of one of our protein of interest. But that was in collaboration. So I have not used the instrument myself, but we collaborated with somebody who had the instrument, and they worked for us in collaboration, and we got a structure solved.
Excuse me, sir.
Okay, bolo.
Sir, what is the sample cost for this cryo?
Yeah, this sample cost. You see, in India, I told you that there is only one Ravi machine in ISC Bangalore or NC, most likely NCPS Bangalore. Okay, so I have never checked their website whether it is even open for public or not. In the sense, can anybody send sample there or not, or whether it is only for the institute samples. Okay, so I am not aware. You can definitely Google it and search it, but when I did, it was a collaborative project. Okay, so we did not have to pay anything. It was a collaboration, so they did for collaborative work.
Okay. Sir, one more question in this regard. Like
Mm.
while doing the sample preparation with the grid,
Mm.
how do you maintain the temperature of that much less? How is it means?
So grid is normally immersed in liquid nitrogen. Okay, so it is already at several in the minus several degrees in negative. So and also as soon as you put your protein sample on that, it is instantly frozen, and then again grid is put back in liquid nitrogen. So it is frozen till it is analyzed by electron microscope. So there is no step in between that where your protein sample will be thawed. Okay, it was instantly frozen and it will remain frozen throughout the processing.
Sir, that I understood, sir. Means we can keep in the liquid nitrogen, but while during the process in electron microscope, you
Hmm.
need to bring it and keep in the electron microscope. Yeah,
So,
and the
so, so,
coating is
so,
also
so,
required.
see again, I'll tell you. So this is not a classical electron microscope. In cryo-EM, it's a modified way of electron microscopy. I have not, as I said, I have not done myself. I have not seen the instrument also. It was all collaboration only. But
Okay.
definitely, it cannot, it's a sample. If it comes to again room temperature, it will defeat the purpose. Okay. So it is modified in such a way that it can analyze the sample in frozen form itself.
Yes, sir. That was the my actually I was interested that to how to process that one
Yeah,
because
definitely. It is a modified way of electron microscope in which the samples are analyzed in frozen form only. They don't come to room temperature at all.
yeah. Actually, because while processing in electron microscope, we have to generally coat with the gold plated.
Yeah,
Sputtering is
so,
required.
here it is not so. Okay, so and also definitely you can go into details of how cryo-EM actually works, but definitely the sample has to be frozen. It has to remain frozen.
Okay. Okay. Thank you very much, sir.
I think
So,
that was also the last slide. So again, I am open to questions. Any question regarding yesterday's presentation or today's presentation? I am open to taking questions.
sir, I would just wanted to ask that it is NCBS or IASC, sir? Which one actually?
99% NCBS.
NCBS. Okay. And can you tell me the professor name, sir?
No, it is not. It is a institute facility. Okay, not a particular person has bought. Okay, it's an institute facility.
Okay. Okay.
So you can just search cryo-EM NCBS or cryo-EM India. It will take you to the desired website.
Okay.
कराना है क्या हनुमंत सर?
नहीं सर, जानकारी लेना है सर, जरूरत पड़ जाता है ना।
बिल्कुल बिल्कुल बिल्कुल, कुछ जगह तो होने चाहिए।
ये सर, नहीं ये क्या है कि सर मैं इलेक्ट्रॉन माइक्रोस्कोप में काम किया था, मेरा कुछ
अच्छा।
सैंपल्स वगैरह किया था, तो उसमें क्या है कि गोल्ड स्पटरिंग करनी पड़ती है, तो मैं थोड़ा सा